The overall goal of this procedure is to extract high-purity RNA from plant seeds.
This method can help answer key questions in the seed biology field, such as understanding gene profiles containing low-level transcripts in seeds. The main advantage of this technique is that researchers are able to prepare high purified RNA with a slight modification to the spin column-based method.
Generally, individuals new to this method will struggle because the high amount of oils, proteins, carbohydrates, and polyphenols in plant seeds make it difficult to isolate highly purified RNA. Demonstrating the procedure will be Kazumi Hikino, a technician from our laboratory.
To begin the experiment, add 1% weight per volume molecular biology grade PVP to cell lysis buffer for RNA extraction and vortex the mixture vigorously. Incubate the PVP for 20 minutes at 25 degrees Celsius to dissolve it completely. After 20 minutes of incubation, mix the buffer gently by turning the tube upside down to prevent bubbles from forming. Harvest fruits from Arabidopsis thaliana plants and place them into two 1.5 mm polypropylene tubes on ice. Then place the fruits on aluminum plates kept at 4 degrees Celsius and isolate seeds from the fruits under a stereo microscope. Next, place approximately 200 isolated seeds into 1.5 ml polypropylene tubes that have been previously stored in an aluminum rack on ice and immediately place the tubes in liquid nitrogen. Remove the tubes from the liquid nitrogen.